Hello and welcome to Nikolai's genetics lessons, and today's problem is a woman with blood type O and Rh positive blood is married to a man with a type A, Mn, Rh negative blood. They had a child. Unfortunately, there was a mix-up at the hospital. Of the 5 children listed below, which one could have been conceived by this couple? And here 5 answers to choose from. As usual, I recommend you to pause video here, try to solve this problem on your own first, and when you would be ready you can run video again and you can compare your answer with my answer and explanation. First of all, here we have 3 blood groups. One is ABO blood group system. This is blood group O of the woman and blood group A of the male. Second blood group we call Mn. So 2 antigens here, M and N. This is proteins, surface proteins, and also Rh factor, which can be present or absent. So how to solve this problem? And actually it is very easy. We just have to break one problem to 3 small problems. First, let's consider ABO blood group system. So a woman belongs to the blood group O, and blood group O has only one genotype. So blood group O can be made by genotype which is O. No any other variants. And male belong to the blood group A, and blood group A, this phenotype can be made by 2 genotypes, A and AO. And of course we are going to get more variants if we cross blood group O, this genotype, with genotype AO, because if we cross with genotype AA, it is easy to predict that 100% of the progeny would also belong to the blood group A, because A allele is dominant over the O allele, which is recessive to allele A and B in the ABO blood group system. But if we cross genotype OO and AO, so we are going to get following results. So O on top and AO on the side. So when we build a simple Punnett square, we can predict what possible outcomes of such a cross. A or here, A or here, or here, and or here. So once again we choose these 2 genotypes because these 2 variants would give us more variation in the progeny. If we would choose this one, 100% result of such a cross would be blood group A. But if we would take these 2, as you see, 50% of the progeny would belong to phenotypic blood group A, and another 50% of the progeny would belong to the phenotypic blood group O. So basically right away we can cross out variant A, where we have AB blood group in the progeny, so this is not going to be our answer. We can cross out variant B, blood group B is not possible in the progeny of this couple. We can cross out blood group AB, this is answer D. And now we basically have to choose between answer C and E. So let's consider next blood group, which we call Mn. So female belong to the blood group N, and male belong to the blood group Mn. In this system we have 2 alleles, allele M and allele N. And 2 alleles in diploid organism can produce 3 genotypes. The first one would be M. second one would be M, N, and the third one would be N, N. So this is 3 genotypes that is made by 2 alleles. And phenotypes would be as follows. Genotype MM would make phenotype M. Genotype MN would make phenotype MN. And genotype NN would make phenotype N. So as you see, this is also very easy and simple. Now if we know that mother belongs to the blood group N, to this phenotype, we know that her genotype is NN. And when we know that male belongs to the blood group MN, we know also genotype of the male, which is MN. Once again, when we build a Punnett square, simple Punnett square, so here is the genotype of the mother of the child, and on the side genotype of the male, MN. Once again, when we build simple Punnett square, we can find probable genotypes and phenotypes of the progeny. So here we have MN, MN here, N here, and N here. So once again, 50% of the progeny would belong to the blood group MN, and another 50% would belong to the blood group N. So we can cross out answer C. Basically, we can stop here and we can say that answer E is the correct one, but let's check if this couple, when female belong to the Rh positive blood group and male belong to the Rh negative blood group, may have Rh positive child, and what are the chances. So basically in this system, Rh positive and negative, we also have 2 alleles, and one allele we just designate with positive sign or plus, and another one with negative sign or minus. Basically, why in this system, instead of letters, we are using positive and negative sign. Positive basically means that this surface protein is present on the surface of the red blood cells, and negative sign means that this protein is absent. So there are also 3 possible genotypes when we have 2 alleles in the gene pool. So 3 possible genotypes would be as follows. Whether we can have 2 alleles, because we got our half of our genome from mother side, another half from our father side. It is possible that we can get 2 alleles that would be normal, but also another situation can be when, for example, from one parent we can get a normal allele, another allele can be defective, and a protein wouldn't be formed. And also we can have such situation when we will get 2 alleles that would be defective. So in this case, these 2 genotypes means Rh positive phenotype, and the last genotype means Rh negative. So once again, 2 alleles can produce 3 genotypes. These 2 genotypes means one phenotype, and this one means another phenotype which would be Rh negative. So if mother Rh positive, she can belong whether to this genotype or to this genotype. If father Rh negative, he only can belong to this genotype. And of course, these 2 genotypes may produce more variation than if we would consider this variant for the mother and this variant for the father. It is easy to predict that 100% of the progeny would be heterozygous, would be positive and negative, and phenotypically would be positive. But if mother would be phenotypically positive, Rh positive, so let's put her genotype here on the side, but father would be Rh negative, so two alleles that is defective. As you see, when we cross these two genotypes in the progeny, we may have following variation: positive, negative here, positive, negative here, negative, 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 negative here. So this cross would produce 50% of the progeny that would be Rh positive, as you see, and another 50% would be Rh negative. So in progeny we may find both positive and Rh negative variants. So as you see, according to two variants of the phenotypes of the mother of her blood group and phenotype of the father, it is possible that the children would be blood group O, would be blood group N, and Rh positive. So this is going to be our answer today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any, and see you in the next video. Goodbye.